Welcome to the Veterinary Research Channel. Today, we're diving into a fascinating study that sheds light on the consistency of bacterial culture and antimicrobial susceptibility testing across different veterinary laboratories. This is crucial for veterinarians because accurate and reliable lab results are essential for effectively treating common infections in our furry patients. The study, titled Comparison of Aerobic Bacterial Culture Among 4 Veterinary Microbiology Laboratories from Dogs with Superficial When testing Staphylococcus intermedius with broth microdilution and disc diffusion, high specificity was observed, but sensitivity varied considerably. Laboratory variability is another critical factor. Earlier studies have demonstrated that differences in inoculum size, incubation time, temperature, and other environmental factors can affect the consistency of results. One study even found a 55% variability in the interpretation of minimum inhibitory concentrations, MIC, when testing a single strain of Staphylococcus aureus across 10 different laboratories. This highlights the need for standardized laboratory practices to minimize variability. Given these concerns, the researchers in the current study aim to evaluate the culture and susceptibility results of a single, uniform sample submitted to 4 different veterinary laboratories. They hypothesized that the results would show high agreement, reflecting the standardized practices now common in larger veterinary labs. Here's how the study was conducted. Dogs with clinical signs of superficial or deep Pyoderma were recruited. To ensure uniformity, samples were collected from the lesions using sterile swabs, which were then rinsed in sterile phosphate-buffered saline and vortex to create a homogeneous bacterial solution. 4 sterile culture swabs were then dipped into this solution in a randomized order and sent to 4 different laboratories: IDEXX, Laboratory 1, Zoetis, Laboratory 2, Antech, Laboratory 3, and Iowa State University Veterinary Diagnostic Laboratory, Laboratory 4. The researchers collected data on several key factors, including the date of submission, the date the results were finalized, the presence and quantity of organisms detected, the specific Staphylococcus species identified, and the antimicrobial susceptibility testing results. All laboratories followed Clinical and Laboratory Standards Institute (CLSI) guidelines for antimicrobial susceptibility testing. Now, let's delve into the results. The researchers found a statistically significant difference in the turnaround time for finalizing results among the 4 laboratories. Laboratory 3 was notably slower, with a median turnaround time of 8 days, compared to 6 days or less for the other laboratories. This difference may be due to Laboratory 4's ability to process samples within the hospital, eliminating the need for shipment. In terms of identifying Staphylococcus species, the laboratories showed fair agreement overall. Staphylococcus species were identified in a range of 80% to 100% of samples across the labs. Interestingly, Laboratory 4, which processed samples on-site, detected Staphylococcus species in 100% of the cases, suggesting that the proximity to the lab and prompt processing may increase detection rates. Staphylococcus intermedius was the most commonly isolated species. However, Laboratory 3 identified a different Staphylococcus species in 25% of the cases when compared to the other laboratories. This discrepancy raises concerns about the potential for misidentification and inappropriate antimicrobial selection. When it came to the quantity of Staphylococcus species detected, there were statistically significant differences among the laboratories. Laboratory 4 reported statistically higher quantities of Staphylococcus species, further supporting the idea that on-site processing may enhance bacterial detection and growth. Perhaps the most critical aspect of the study was the comparison of antimicrobial susceptibility testing results. The researchers focused on antimicrobials commonly tested by all 4 laboratories, including oxacillin, amoxicillin clavulanic acid, cephalexin, cefovecin, cefpodoxime, amikacin, gentamicin, enrofloxacin, marbofloxacin, clindamycin, doxycycline, chloramphenicol, and trimethoprim sulfamethoxazole. They found that agreement among the laboratories ranged from good to very good for all antimicrobials tested. Specifically, there was very good agreement for oxacillin, amoxicillin clavulanic acid, cephalexin, cefovecin, cefpodoxime, amikacin, marbofloxacin, and trimethoprim sulfamethoxazole. Agreement was good for gentamicin, enrofloxacin, clindamycin, doxycycline, and chloramphenicol. This level of consistency is encouraging, particularly for methicillin resistance, which is a major concern in veterinary dermatology. The study's findings have several important implications for veterinary practitioners. First, while there is generally good agreement in antimicrobial susceptibility testing across different laboratories, variability exists, particularly in turnaround time and species identification. Therefore, it's essential to consider these factors when selecting a laboratory. A faster turnaround time can expedite treatment, while accurate species identification is crucial for appropriate antimicrobial selection. Second, the study highlights the importance of standardized laboratory practices and adherence to CLSI guidelines. These standards help to minimize variability and ensure reliable results. Third, the study suggests that proximity to the laboratory and prompt sample processing may improve bacterial detection rates. When possible, choosing a local laboratory or ensuring timely sample delivery may enhance the accuracy of culture results. Despite its valuable insights, the study has some limitations. The sample size was relatively small, and a larger, multi-institutional study would provide more robust data. Additionally, the study focused on commonly used commercial veterinary laboratories, and including smaller, regionally exclusive labs or university veterinary diagnostic laboratories could offer a broader perspective. In conclusion, this study provides a comprehensive comparison of bacterial culture and antimicrobial susceptibility testing across 4 veterinary laboratories. While mild variability was observed, particularly in turnaround time and species identification, the overall agreement in antimicrobial susceptibility testing was reassuring. The findings underscore the importance of choosing a reliable laboratory, considering turnaround time and species identification accuracy, and adhering to standardized laboratory practices. Thank you for listening to the Veterinary Research Channel, and as always, keep those minds inspired, hearts light, and tails wagging.